Lewis. I'm the founder and CEO of Entos Pharmaceuticals. We're a platform technology gene delivery company based in Edmonton, Alberta, Canada. And happy to tell you today a little bit about our technology and what we're planning for gene delivery in the future. So I always like to start about thinking, the opportunity for genetic medicine. We have, over the past, couple decades, sequenced the human genome, sequenced individual patients, sequenced individual diseases, and developed tools to be able to precisely upregulate, downregulate, and as you saw from the last presentation, precisely edit genes to affect cures in many different diseases. So the opportunity for curing, pretty much all the diseases that affect mankind are within our grasp. But what I would say the key challenge in all of this, despite all this promise, is our ability to deliver these tools safely, effectively, to those cells where they're going to have that therapeutic curative benefit. And so that's what Entos Pharmaceuticals is doing. And so many of us at this conference and in this industry realize our understanding of sort of the tools that are available. And so broadly bucketed, we have viral and non-viral gene delivery technology. And these are the commercialized platforms. So on the DNA side, we have viruses like AAV, which have evolved for millions of years to do an incredibly good job of getting into cells and delivering DNA, obviously for the purpose of replicating themselves, but we've hijacked that to deliver therapeutic and curative cargoes. But AAV is not the perfect delivery system. It's a virus, so it elicits its own immune response, which makes redosing challenging. You have to get the correct dose the very first time. And it has its own challenges in that it can deliver DNA only. It has cargo capacity limitations, particularly for these large genes, and manufacturing at scale challenges. And so non-viral technologies like lipid nanoparticles have been developed to try to address some of these limitations. And we've obviously seen dramatic success, particularly with the COVID vaccines of LNP. But LNP as a systemic gene delivery tool, whether it be for siRNA, RNA, or even DNA, has significant challenges, and that's because the way they're formulated and their mechanism of action relies on endosomal escape. And endosomal escape uses potent ionizable lipids to basically physically break through the endosomal membrane to leak some of the cargo into the cytoplasm. And because of this mechanism, we see dose-limiting toxicities, particularly in the liver, because also the way they're formulated makes them extremely liver-tropic. And so we're seeing for some applications they work extremely well, but for systemic administration for many diseases it's been really challenging to have extrahepatic delivery. And while repeat dosing is possible, there also really haven't been developed for delivery of DNA, and I'll get into that a little bit. Delivery of DNA and LNP creates quite an immunogenic response. And so at Entos we thought, could we combine the best aspects of both viral and non-viral delivery to create a system that can address some of these limitations? And so we've done that by combining a very novel, very unique, very tiny viral fusion protein into a lipid particle similar in concept to an LNP. But what this tiny viral fusion protein does is facilitates direct fusion of a lipid particle with the plasma membrane of target cells, completely avoiding endosomal uptake and escape, and therefore delivering the cargo directly into the cytoplasm. And so we've developed a platform whereby we can encapsulate and deliver RNA, DNA, or even protein directly to cells through a novel mechanism of action. And the key to our technology was a discovery made by Entos co-founder Roy Duncan about 20 years ago, where he discovered the only non-envelope virus to make a fusion protein. So you may be aware, envelope viruses like HIV, like influenza, for instance, express fusion proteins to allow them to fuse with target cells and deliver their payload. But typically these proteins they're pretty hard to work with. They're pretty massive. Multi-subunit, multi-transmembrane domain. They're immunogenic in and of themselves, and they're pretty much very challenging to manufacture, for instance, for a drug delivery system. Roy Duncan discovered this novel class of fusogenic orthoreoviruses that's non-enveloped, but they still express a fusion protein. It turns out this fusion protein is really, really novel, very, very tiny. He called them the fusion-associated small transmembrane or FAST proteins. And he showed way back in 2005 that if you incorporate these FAST proteins into a lipid particle, in this case a liposome, he was able to demonstrate very effective fusion of that liposome with the target cell and distribution of the cargo directly into the cytoplasm. And what's most important is that when he inhibited endocytosis, he was only able to inhibit delivery by less than 20%. So for the most part, this is a completely endosomal escape independent mechanism of delivery. And so he and I, over the past 15 years or so, have been characterizing fast proteins from different orthoreoviruses to create a fully engineered, synthetic, manufacturable version of the fast protein that is in our current clinical formulations. So we call it to distinguish it from LNP, we call it the proteolipid vehicle platform. It's a combination of a lipid particle with this fusion protein. And so because the fusion protein is actually delivering the nucleic acid, we've been able to sort of reimagine what a lipid nanoparticle can be. We're not constrained by having ionizable lipid or cholesterol. And so we've generated panels of formulations to effectively encapsulate and deliver DNA, RNA, protein, combinations thereof. And I spent a lot of time on obviously the safety and tolerability and the ability for these to be distributed throughout the body extrahepatically. And also on the manufacturing. So we've developed a pretty slick, single-step, continuous flow microfluidic process whereby we combine fast protein, lipids, and nucleic acid cargo in a single step. I'm going to spend the next few minutes telling you a little bit about the properties of the platform and how we think we can make a dent in diseases systemically. So first of all, everybody, many people are familiar with LNP. LNP typically have a potent ionizable lipid, roughly half of the formulation, and about 40% cholesterol, and the rest is taken up by a helper lipid and a little bit of pegylated lipid. And so we don't have those constraints. We don't need cholesterol. We need, obviously, some charge to encapsulate the nucleic acid cargo, but we don't require the ionizable lipid for endosomal escape. So we've been able to create these novel formulations of both biodegradable and well-tolerated lipids that are substantially better tolerated than LNP, up to 100x in many situations. And it gives us a lot more flexibility in the way we complex with the nucleic acid components, giving us a lot more flexibility, novel design space around adding novel lipids or incorporating different activities. And so this has had two dramatic effects on the way they behave when you administer them in vivo. I'm showing you data here, either delivering, these are reporter genes, luciferase, either in DNA or RNA. They behave very, very similarly. And you can see when you administer DNA or RNA in an LNP systemically in a mouse, pretty much 85–90% goes to the liver first pass. And these non-cholesterol containing PLVs basically go everywhere. So the liver is about the 6th or 7th organ down in the biodistribution list, and we see uptake in pretty much every cell that we look at. Also very interesting is when we administer locally. For instance, we have a phase 2 vaccine program, intramuscular injection of a DNA-containing luciferase reporter in this case, get really robust expression in the muscle, in the sort of local inflammatory cells and the draining lymph node a little bit, but really no systemic exposure. Similarly for CNS, we inject intrathecally. We have great transit to the CNS, great expression throughout the CNS, but no systemic exposure. And I'll mention this is something we're excited about for the future. Because of this flexibility in formulation, we can create very stable formulations that can survive the GI tract intact, for instance, and so we have the opportunity to develop oral formulations as well. And so obviously, if we can deliver RNA, DNA, protein, we can deliver it to every cell of the body, we have a lot of things to think about as far as a company and how we develop candidates and how we partner. So we've really focused on the past 2 years in developing very specific formulations for areas of high interest. One of these is CNS. So we've developed a very potent, highly tolerable CNS formulation for delivery of RNA or DNA. And we've, in the past 2 years, have developed a relationship with Eli Lilly, developing novel CNS genetic medicines. We've developed an ocular formulation that can be delivered through multiple routes of administration, including subretinally and intravitreally, and we get really nice penetration expression in the multiple cell layers of the retina, for instance, including photoreceptors. We have a very nice lung formulation, so systemic administration of a formulation that has almost zero liver exposure and very, very strong lung exposure to various cell types. And also a muscle formulation. So obviously there's debilitating rare diseases in muscle. And so we have a systemically administered muscle formulation that can deliver DNA or RNA systemically and have good uptaking expression in muscle. So that's biodistribution. So this slide is going to be a little bit surprising. but and it's probably the reason why you don't see many LNP-delivered DNA programs. So, and this surprised us as well. So the other impact the reformulation has on the PLV is systemic tolerability when delivering DNA in particular. I'm showing you the results from 2 experiments here. We systemically delivered DNA at different doses in a PLV or in an MC3-based LNP. And we actually saw dramatic and surprising toxicity with the LNP. So at 1 and 5 mg per kg, basically all the animals died. You can see here the livers are highly hemorrhagic. The PLV, even at 20 mg per kg, livers are completely clean, animals are completely fine. We redid this experiment where we lowered the MC3 LNP DNA dose to 0.5 mg per kg and looked at systemic inflammatory, pro-inflammatory cytokines at 24 hours, and you can see there's a dramatic difference here. So IL-6 in the LNP group is up 100,000-fold. We see huge spikes in the inflammatory cytokines, and the PLV, even at 20 mg per kg, is not statistically significantly above the saline-injected group. So we seem to, because of our novel mechanism of action, be completely avoiding the immunogenicity associated with DNA. And obviously, we're using a viral fusion protein. So the first question is, are you messing with your redosing ability? Are you creating an immune response? And so we've tested this extensively. I'm just showing an example of a couple experiments, whether we locally or systemically dose mice repeatedly, once a month for 5 months, or we treated 11 monkeys, NHP, at 6 mg per kg, a huge dose twice. And we have a GLP-validated anti-fast protein ELISA. And basically, in every animal, we're getting a nominal response. We got a couple animals in the IV-injected group that are just above lower limit of detection, but nothing remotely close to any neutralizing level. So we're basically able to repeat dose, and we don't see any neutralizing activity. So the one thing we've really focused on is optimizing PLV formulations that can deliver DNA effectively throughout the body. So, it comes to mind many different applications, including, gene editing with large gene replacement, for instance, or protein replacement. And so in this case, I'm showing an example of a delivery of a plasmid DNA expressing luciferase reporter delivered in a single dose systemically. And we're seeing we're going back 2 months later and doing immunohistochemistry for luciferase, basically every cell of the lung, liver, and spleen is expressing the reporter. So very good tissue penetration and long-term expression of a long-term episomal plasmid. I'll just finish the talk by talking about one of the programs we're really excited about with a partner, Oshin Biotechnologies. This program is really focused on, and this is obviously a big topic right now with GLP-1 inhibitors, the preservation of lean mass, but preservation of lean mass is particularly important as we age. And so follistatin is a well understood TGF-beta family secreted factor that contributes significantly to muscle growth. And we created a single dose, systemically administered DNA formulation of follistatin and tested this in cohorts of mice. And basically with a single systemic dose, we're seeing, after a year, we're seeing the mice, roughly 50% increase in weight and a roughly doubling of their muscle strength, which is in parallel with substantial increased muscle fiber, cross-sectional area, and size. So we're really excited about applying this PLV technology to many other systemically administered gene therapies. So as far as a company, we've been very active in external partnerships. I mentioned our fantastic relationship with Eli Lilly and Oshin Biotechnologies. We have our furthest along clinical program is actually a COVID-19 DNA vaccine that we just finished phase 2. We're going back to clinic as a booster in a couple months. We just announced last week a great relationship with the Bill and Melinda Gates Foundation, working on intramuscularly injected therapeutics that will produce neutralizing antibodies for HIV and malaria. Really excited about that relationship and the application of that approach to many other diseases as well. Internally, as I mentioned, we have a program on metabolic disease that you're happy to approach me and talk about later. But as I mentioned, we're focusing on ophthalmology, lung delivery, muscle delivery, as well as our clinical DNA vaccine program. I'll finish it up there. So I'm here at the meeting. I'm actually joined by Arun Maturi, our CSO, and Jason Ding, our Chief Business Officer, is here. So feel free to approach us or get in touch if you want to know more about the platform. And thank you very much.